Hello friends, welcome to my channel Easy Life Science. Here I am talking about important questions, their answers with detailed explanation for set exam subject Life Science part 9. So please subscribe my channel Easy Life Science, like my video, share it to your friends, and don't forget to press the bell icon to get the notification of my newly uploaded video. So let's start. Starting from the question number first, match the plant part in a group first with the product obtained in a group second and a representative plant species in a group third in a correct combination. You have to find out a correct combination from options. So the first, there is a bark. The bark of a plant Cinnamomum giranicum. In our Hindi language, this plant is known as a dalchini. Cinnamomum giranicum. This plant known as a dalchini in a Hindi language, it belongs to a family Lauraceae. Cinnamon oil is a aromatic liquid obtained from the twigs, bark and leaves of this plant, and this bark is used as for a preparation of spices. So the bark, aromatic oil producing plant, that is a Cinnamomum giranicum, and bark. Then a leaf. Camellia sinensis is a common name for a tea plant. Its leaves are used as a tea powder. So Q to the tannins and Camellia sinensis. Then the flowers of Crocus sativus. Its common name is a kesar in a Hindi language, or a kumkum, and in English it is a saffron. This plant is a flowering plant from a family Iridaceae. The floral stigma are used as a saffron. So the floral stigma of a saffron, Crocus sativus, this is a combination. Then the fruits of a Papaver somniferum, or a coden. Coden is a opioid and prodrug of a morphine, mainly used to treat a pain, coughing and a diarrhea. It is found in a sap of opium poppy. The opium poppy in a Hindi is known as a afim, and in Marathi it is a afu. So fruit of opium poppy or a Papaver somniferum, it is a source of a coden. So this is a combination, and for this combination the option 4th is the answer. So moving to the next question. Match the following ecological terms with their appropriate definitions. We know that niche. The niche is a place of living organism in biotic environment and its relation to food and enemies. Or we can say that a niche is a physical habitat of the organism. Then the biota. The biota is a totality of organisms or a totality of a flora and fauna in a particular place or a region. The biota comprises all living organisms, including bacteria, fungi, plants, animals that inhabit a given area. Then a trophic level. The trophic level a position of a species in a food chain. Then habitat. The habitat is a physical environment of the organism. So this combination is in option B. Option B is the answer of this question. Moving to the next question, match the following alkaloids with their uses and source of plant in correct combination. Alkaloid use and a source plant. The first alkaloid is a morphine. The morphine, the source plant for a morphine is a Papaver somniferum or a opium poppy, and this has been used medicinally for 5,000 years back. It is a source of three important alkaloids including morphine, codeine and papaverine. These alkaloids are used as analgesic. Analgesic substances are the substances that reduces the pain. So the P, that is a morphine, analgesic, and a source for this plant is a opium poppy, that is a Papaver somniferum. This is a combination. Then quinine, quinine and other cinchona alkaloids including quinidine, cinchonine, cincodyne are the effective against malaria. So these are known as anti-malarial. These are obtained from Cinchona officinalis. So quinine, it is anti-malarial. Source plant, that is Cinchona officinalis. Then the atropine. The atropine is produced by or a source plant for this Hyoscyamus niger, or Hyoscyamus niger is a source plant for the atropine. Hyoscyamus is an isomer of atropine and is also the precursor for the synthesis of scopolamine. This atropine is obtained from leaves and seeds of a plant species, that is a Hyoscyamus niger, commonly known as a henbane. It is a poisonous plant from a family Solanaceae. The alkaloid atropine is anticholinergic. The anticholinergic are the drugs that blocks and inhibit the activity of neurotransmitter, that is acetylcholine. So atropine, it is anticholinergic, and the source for this plant is Hyoscyamus niger. Then the vinblastine. The vinblastine is an anti-cancer agent commonly obtained from a tree, that is a Catharensis rosus, commonly known as a periwinkle, or in Hindi Sadabahar, or in Marathi Sadafuli. It is a species of flowers from plant family Apocynaceae. It is an ornamental and medicinal plant. So vinblastine to the use anti-cancer, source plant, that is a Catharensis rosus. So such combination is in option A. So on answer of this question is option A. So next question, some selection markers and the corresponding genes used in a plant genetic engineering are given below. Choose the correct combination, selection markers and their genes. The first, that is a NPT second gene. The NPT second gene codes for aminoglycoside phosphotransferase, carrying or giving a resistance to an antibiotic such as kanamycin and gentamicin. So this matches with the kanamycin. Next, that is a NPT fourth gene, and this gene is for resistance toward the hygromycin. The BAR gene, BAR stands for a bilapose resistance gene, which is isolated from Species Streptomyces hygroscopicus. So BAR for resistance toward the bilapose. PMI gene, a selectable marker adapted for agrobacterium mediated transformation. This gene is encoded for phosphomannose isomerase and this enzyme convert a mannose 6 phosphate to fructose 6 phosphate, and only transformer cell will capable of utilizing mannose as a carbon source. So PMI gene is a selectable marker for a mannose. So this combination is in option C. Option C is the answer of this question. Moving to the next question, choose the correct taxonomical hierarchy among the following. The hierarchy is asked here in this question. Start from a species, genus, family, species, genus, family, order, class, phylum, and domain. This is a taxonomical hierarchy for a classification. Option A is the answer of this question. Moving to the next question. The technique of pure culture or a technique of microbial pure culture was pioneered by who discovered the pure culture technique? The technique of pure culture was discovered by a German scientist Robert Koch, who also discovered the causative agent for a deadly diseases like tuberculosis, cholera, and anthrax. He discovered solid culture media and was the first to obtain pure culture of a bacteria. So answer for this question is the option D, that is Robert Koch. The another option, that is Edward Jenner. The Edward Jenner developed the first vaccine. This was a vaccine against a smallpox. Louis Pasteur, he was the first to demonstrate that the infectious diseases are caused by microbes. He disproved the concept of a spontaneous generation. Louis Pasteur developed the process of pasteurization. Then the Robert Hooke, he gave the proof for existence of a microscopic organism. So option D is the answer of this question. Robert Koch gave us a technique of a pure culture. Then next, the antibacterial agent, that is a trimethoprim, is an inhibitor of. The antibacterial agent, that is a trimethoprim. Trimethoprim is used to treat and prevent a urinary tract infection such as cystitis. Trimethoprim exerts antimicrobial activity by blocking the reduction of dihydrofolate to tetrahydrofolate. The tetrahydrofolate is the active form of a folic acid. So the trimethoprim is a inhibitor of the enzyme dihydrofolate reductase. The next question, which one of the following condition favors maximum expression of lac-operon genes in E. coli? To express the maximum gene of a lac-operon, there should be a low amount of glucose, high amount of cyclic AMP, and high amount of lactose. Because the cyclic AMP is a positive regulator for the lac-operon, lactose is an inducer, and glucose is sometimes it is an inhibitor for a lac-operon. So in a medium there should be a low glucose, high lactose, and high cyclic AMP. Then match the cellular organs in a group first with its function in a group second. A Golgi apparatus, it is a post box of a cell. It transport proteins and other material to a particular destination in a cell. So it is a protein sorting organelle in a cell, or it is a post box in a cell. So it match with a protein sorting. Nucleolus, the nucleolus having a role in ribosomal RNA synthesis. Peroxisomes, where the lipids are degraded, and the proteasome, the proteins are degraded in a proteasome by a ubiquitination process. So this combination is in option B. Option B is the answer of this question. Then moving to the next question, you can take the screenshot of a lac operon. Moving to the next question, which one of the following will increase the resolution of a light microscope? The resolution means the minimum distance in between two objects that the eye can see that these two objects are different or separated. Here the question is asked, which one of the following will increase the resolution of a light microscope? The option A saying that decreasing the numerical aperture of the objective lens. Because of this, decreasing a numerical aperture, the resolution will not get increased. Using an objective lens with a longer working distance, it is not increasing a resolution of light microscope. Using a medium of a higher refractive index, yes, by using a medium of a higher refractive index, the resolution of a light microscope can be increased, and for this purpose, at time of using a light microscope, the medium of a higher refractive index is used. Option D, increasing the wavelength of light. Increasing a wavelength of light, it decreases a resolution power. So option C is the answer of this question. This question is repeated. Which one of the following conditions favor the maximum expression of a lac operon in E. coli? It can be increased by a low glucose, high cyclic AMP and high lactose. Then the next question, which one of the following disease is treated by a neuroamylase inhibitor? The neuroamylase inhibitors are a class of drugs which blocks the neuroamylase enzyme. They are commonly used antiviral drugs against influenza. Viral neuroamylases are essential for influenza reproduction and facilitating viral budding at time of reproduction. Viral budding from a host cell is necessary for a lytic cycle. The neuroamylase inhibitor includes the drugs like oseltamivir, paramivir, Tamiflu and Relenza. These are some neuroamylase drugs or neuroamylase inhibiting drugs. So they are used to treat the diseases like influenza. Option C is the answer of this question. Next, which one of the following does not provide the three-dimensional image? The atomic force microscopy, confocal scanning laser microscopy and differential interference contrast microscopy, these all provides the three-dimensional structure of the image or three-dimensional image, but the phase contrast microscopy does not provide a three-dimensional image. So option D is the answer of this question. Then the next question, which one of the following is not a site for in situ conservation? In situ conservation means the conservation of a species in its natural habitat and the maintenance and recovery of viable population of a species in their original place. It includes biosphere reserves, national parks, wildlife sanctuary. The example of in situ conservations are biosphere reserves, national parks, wildlife sanctuary. So here question is asked, which one of the following is not a site for in situ conservation? In situ conservation means conservation of a species in its natural habitat, biosphere reserves, wildlife sanctuary and biodiversity hotspot. These all are in situ conservation, but the zoological garden is not in situ conservation, it is a ex situ. Ex situ conservation means it is a conservation of a living species in the artful habitat or rather than their natural habitat. It includes botanical garden, zoological garden, cryopreservation, DNA banks, aquarium and zoo. So option C is the answer of this question. Which one of the following is the precursor molecule for a corticosteroids? Corticosteroids are steroid hormones produced by adrenal cortex, including glucocorticoids and mineralocorticoids. The progesterone is a precursor for corticosteroids. The function of a corticosteroid is these steroids acts by binding to intracellular receptors, which then acts to modulate the gene transcription in a target tissue. So here the question is asked, which one of the following is the precursor molecule for a corticosteroid? The precursor molecule for corticosteroid is a progesterone. Option C is the answer of this question. The next question, which one of the following leukocyte is a phagocytic and has a clear cytoplasm? Leukocytes are parts of a body's immune system. They help the bodies to fight against infection. The types of leukocytes are granulocyte, that is eosinophils, basophils and neutrophils, and agranulocyte like monocyte and a lymphocyte. The monocyte are the leukocyte which are phagocytic in nature and has a clear cytoplasm. Just have a look in cells in immune system. The neutrophils, these are multi-lobed cells in our blood and they gives protection against bacterial and fungal infection, and these are the most common first responders to microbial infections. They are multi-lobed. Then eosinophils, these cells are bilobed like this and they gives a protection against a parasitic infection and allergic reaction like inflammation and atopy. Then the basophil are maybe bilobed or trilobed. They gives a response to allergic and at time of infection they release a vasodilating substance. Lymphocytes, after staining they appears as a deep stained, includes B cells, CD4 plus cells or helper cells, CD8 plus cell or T cytosol cells, and operates primarily in the lymphatic system. And a monocyte, our question is on a monocyte. Monocytes are kidney shaped cells, carry out a phagocytosis of pathogens, presents the antigen to T cells. Eventually they becomes tissue macrophages which removes dead debris and attacks the microorganisms. So the answer of this question is a monocyte. Then moving to the next question, which one of the following technique can be used for detecting the subcellular localization of serotonin receptor in intact cell? Here the question is asked on localization of intracellular receptors, and one technique is there, that is immunoelectron microscopy. The immunoelectron microscopy is used to localize molecules at the ultrastructural level by labeling them with a specific antibody. So answer for this question is option A. Moving to the next question. The round dance is performed by farragal bees to indicate the distance between a food source and their colony. Which one of the following best represents this distance? The question is on bees. For bees, Their forage or a food supply consists of a nectar and a pollen from blooming plants within a flight ranges. Forager bees function not only as gathering of a food for their colony but also as sensory unit shaped by a natural selection to gather information regarding to location of a forage site. These bee or a forager bee transmit this information to a colony members by means of a waggle dance, and the waggle dance is carried out at a distance of 45 meters. So answer of this question is 45 meters, option A. Then moving to the next question, which one of the following phyla have a cynocytes? Cynocytes are specialized cells that have a single flagellum surrounded by a net-like collar of a microvilli. Cynocytes joins together creating a cyanoderm where they perform two major functions. The first is to create a flow of water and the second is to capture a food atoms. The phylum Porifera have a cynocytes. So option D is the answer of this question. Then next question, which one of the following glial cell is not derived from a ectoderm? Four glial cells are there: astrocytes, microglial cells, oligodendrocyte and ependyma. The ectodermal original glial cell is astrocyte, oligodendrocyte and ependyma, but the microglial cells are not derived from ectoderms. So option B is the answer of this question. Here at time of the formation of germ layers, three germ layers are formed: ectoderm, mesoderm and endoderm. Ectoderm is an external layer, mesoderm is a middle layer and endoderm is an internal layer, and these layers give rise to a formation of a number of cells. Ectoderm is one of the three primary germ layers formed in early embryonic development. It is the outermost layer and it is superficial to a mesoderm. The mesoderm is a middle layer. The innermost layer is a endoderm. The ectoderm leads to giving rise to a kin cells of a epidermis, neurons of a brains and pigment cells are formed from this layer. Skeletal muscles, smooth muscles, cardiac muscle, tubules of a kidney and RBC are formed from a mesoderm, whereas a pancreatic cell, alveolar cell of lungs and thyroid cells are rising from a endoderm. Next question, the genome of both human and Drosophila code for an amylase that acts on the same substrate. However, the sequence of nucleotides in the gene coding the two is dissimilar. This is an example of which one of the following type of evolution, and this kind of evolution is a convergent evolution. Next question, transitional epithelia is found in which one of the following organ? The transitional epithelium of urinary tract is lined by a layer of glycosaminoglycans that functions to prevent microbial and cystal adherence to the bladder of a epithelium. So transitional epithelia are found in a urinary bladder. So option D is the answer of this question. Moving to the next question, visual transduction cascade is activated by a rhodopsin and involves a degradation rather than a synthesis of which one of the following second messenger molecule? Here rhodopsin is a membrane protein that detects light in the rod photoreceptor cell. Like other G protein coupled receptor or a GPCRS, rhodopsin exists in a equilibrium between its activated and inactivated form. Activated rhodopsin catalyzes replacement of a GDP by a GTP on alpha subunit of a heterotrimeric G protein. So option C, that is a cyclic GMP, is the answer of this question. In the moving to the next question, match the following components listed in a column first with their respective organ in column second. Column first, that is a endolymph. The endolymph is also known as a scarpa fluid, is a clear fluid that can found in a membranous labyrinth of the inner ear. Fibrous humor. The fibrous humor is a transparent, colorless gel-like substance located in the posterior chamber of the eye, and it helps to maintain the round shape of our eye and can also help to vision clarity and shock absorbance. Endolymphs for ear, fibrous humor for eye, vas deferens. It is also called as a spermaduct. The spermaduct, job of this duct is to remove or to move the sperm away from its storage place in the testicle. This duct is coiled tube that carries a sperm out of the testes. Corpus luteum. The corpus luteum is a normal cyst that forms on the every single month in a woman of childbearing age. This cyst is actually a group of cells inside a ovary that forms during each menstrual cycle. It appears right after an egg leaves your ovary. At a pregnancy, the corpus luteum secretes a progesterone for preparing the uterus for implantation. So option A is the answer of this question. Moving to the next question, match the following genetic disorder in a column first with associated typical chromosomal changes mentioned in a column second. The favorite question of a set exam. Multiple times the questions are asked on this genetic disorders. The first, Klinefelter syndrome. It is a sex chromosome disorder in boys and in men. This disorder occurs. It results from the presence of an extra X chromosome in a cells. People typically have a 46 chromosome in each cell, 2 of which are a sex chromosome. Females have 2 X chromosome, that is a 46XX, and males have a 1X and 1Y chromosome, that is a 46XY. But in a Klinefelter syndrome of boys and men, there is a extra copy of a X chromosome, that is a 46XXY or a 47XXY. Then the Down syndrome. Down syndrome is also known as a trisomy of a 24th of a chromosome, not 24, 21. It is a trisomy of a 21 chromosome. It is caused by a presence of third copy of a chromosome number 21. It is usually associated with a physical growth delay, intellectual disability. So Down syndrome matches to the trisomy of chromosome number 21. Then the Turner syndrome. Turner syndrome is also known as 45X or a 45XO, is a genetic condition in which a female is partially or completely missing an X chromosome. Symptoms are short and waved neck, swollen hands. So the Turner syndrome, 45XO. Then next, Cryo-Chart syndrome, also known as a 5p- syndrome. It is a chromosomal condition that results when a piece of a chromosome number 5 is missing. Infants with this condition often have a high pitched cry that sounds like that of the cat. So the name for this disorder is a Cradock syndrome. The piece of a chromosome number 5 is missing. So 5p minus. So another some diseases are there like Edwards syndrome. The Edwards syndrome is also known as a trisomy of chromosome number 18. It is a genetic disorder caused by a presence of third copy of a chromosome number 18. Symptoms are hurt depicts small head and intellectual disability. So answer for this question is option D. Moving to the next question, match the household insect factors in a column first with their associated disease in a column second. Some household insects are given in a column first and their diseases are given in a column second. You have to match it. The first, that is a kissing bug, that is a Hemiptera. The kissing bugs are also called as tritomine bugs. These blood-sucking bugs transmit the Trypanosoma cruzi by biting an infected animals or a human. Trypanosoma cruzi is a parasite which causes a disease, that is a Chagas disease, to animal and humans. This disease, that is a Chagas disease, is also known as trypanosomiasis. So kissing bug Hemiptera matches to the Chagas disease or a trypanosomiasis. Then a sandfly or a diphtheria. This causes a Kala Azar or a leishmaniasis. The Kala Azar or leishmaniasis are a group of diseases caused by a protozoan parasites like Leishmania pestis. These parasites are transmitted to a human by bite of an infected female phlebotomine sandfly. Then deer fly or a diphtheria. Deer fly ulceroglandular. This is the most common form of a tularemia, occurs by bite of ticks or a deer fly. A skin ulcer appears at the site where the bacteria entered into the body. The ulcer is accompanied by a swelling of a regional lymph gland. So deer fly, tularemia. The deer fly, scientific name for this is a Chrysops species. Then oriental rat flea, Cyprinoptera. Oriental rat flea or a Cyprinoptera, also known as a tropical rat flea, is a parasite of rodents and a vector. They are causative agent for bubonic plague. Bubonic plague. The bubonic plague is caused by a Yersinia pestis. Scientific name for oriental rat flea is Genopsylla chuopsis. Genopsylla chuopsis. So this combination is in option D. Option D is the answer of this question. The next question, match the following subcellular organisms in column first with associated function in column second. Nucleolus. Nucleus for ribosome biogenesis or ribosome synthesis. Peroxisomes, oxidation of fatty acids and amino acids. ER, glycoprotein biosynthesis, and Golgi body protein trafficking. So option C is the answer of this question. Then the next question, in relation to agrobacterium mediated genetic engineering in a plant, match the following correct combination. Some genes are given and their function are given there. So match them. First, that is a FIR-A gene. Agrobacterium mediated gene transfer results in the integration of foreign genes at a single locus in a host plant without associated vector, also known to produce a marker free plants. Agrobacterium perceives a plant derived signals to activate its virulence gene, which are responsible for transferring and integrating its transporter DNA, that is a tDNA, from its tumor inducing plasmid into a plant nucleus. The Vir genes of Agrobacterium tumefaciens tumor inducing plasmid directs the transfer of oncogenic portion of a TI, that is a tumor inducing plasmid, that is transferred to a plant cell. The Vir A and Vir G genes products are required for regulation of Vir regulon on tumor inducing plasmid of Agrobacterium tumefaciens. The Vir A, it has a function as a acetylsyringin receptor. Vir B for a conjugational tube formation, Vir D1 for a topoisomerase time of DNA replication, and Vir G for regulon for all other Vir operons, or it is a inducer for all other Vir operons. This is in option C. Option C is the answer of this question. Here in this image, have a look on the virulence gene of a Vir operon, Vir A to Vir G. 6 essential operons are there, Vir A, B, C, D, E and G, and 2 non-essential are there, Vir F and Vir H, and their functions are given in this image. So take a screenshot of this, because to next time the question will be asked on remaining these Vir genes. Then next, match the following digestive enzyme in a column first with their respective function in column second. First, erepsin. Erepsin is a mixture of enzyme contained in a protein fraction found in intestinal juices that digest peptones into amino acids. And erepsin is produced and secreted by the intestinal glands in a ileum and the pancreas. So it converts a polypeptide into amino acids. Trypsins. The trypsins is also known as a lipase, a digestive enzyme present in the pancreatic juice that helps in the digestion of fats. Trypsin enzyme catalyzes the breakdown of fats into smaller molecules of glycerol and fatty acids. So trypsin matches with it converts a fat into fatty acid and glycerol. Then pepsin. Pepsin is a stomach enzyme that serves to digest proteins found in ingested foods. Gastric chief cells secrete a pepsin as in an inactive zymogen called a pepsinogen. The parietal cells within a stomach lining secrete a HCl that lowers the pH of stomach, and so the pepsinogen converted into a pepsin. The pepsin convert the protein into a peptides. Then enterokinase. Enterokinase is an enzyme produced by cells of a duodenum, and this convert trypsinogen into its active form trypsin. Enterokinase activates a trypsinogen into a trypsin. So option B is the answer of this question. The next question, here in this image, number of enzymes are there and their functions are there. So you can take the screenshot of it. Match the following column first and column second. Some proteins are there and their organs are there. So match it. The first, that is a keratin. It is a structural fibrous protein. Alpha keratin is a type of keratin found in vertebrates. It is a structural material in scales, hairs, nails, feathers, horns, claws, hooves, and outer layer of skin. And keratin produces a outer layer of skin, that is it matches with the sport. Surfactants. Surfactants are largely present in lungs. Surfactants are also called as a surface active agent, substances that reduce the surface tension and thereby increasing the spreading and wetting property. So there is a need of a surfactant in a lungs. Sodium stearate is a good example of a surfactant. It is the most common surfactant in a soap. Then are procarboxy peptidase. The procarboxy peptidase is the inactive precursor of carboxy peptidase, which converted to an active enzyme by the action of a trypsin. The active form of a procarboxy peptidase is a carboxy peptidase. The carboxy peptidase are the protease enzyme that hydrolyzes or cleaves a peptide bond at a carboxy terminal or a C terminal of a protein. So procarboxy peptide is secreted from a pancreas. Then the albumin. The albumin is a protein made by a liver. Albumin enters in a bloodstream and helps to keep the fluid leaking out of the blood vessel into a outer tissue. It is also carries hormones, vitamins and enzymes throughout the body. Albumin is a water soluble globular protein produced in a liver. So this combination is in option C. Option C is the answer of this question. This is a repeated question. So thank you for watching me. Thank you once again. So please subscribe my channel Easy Life Science, like my video, share it to your friends and don't forget to press the bell icon to get the notification of my newly uploaded video. Thank you once again.